Welcome to Easy Microbiology. Good morning everyone. Today we will be discussing Clostridium septicum, an important opportunistic pathogen that can cause various infections, including gas gangrene and septicemia. We will explore its habitat, morphological features, biochemical characteristics, host range, virulence factors, mechanisms of antibiotic resistance, and more. Let's take a closer look and begin. Please like and subscribe to our channel, which will be your great support and encourage us for making educational videos in upcoming days. Clostridium septicum is a type of bacteria that is commonly found in soil and the digestive systems of humans and animals. It is capable of causing infections, particularly in individuals with weakened immune systems. Some of the infections it can cause include gas gangrene, a condition characterized by tissue death and the production of gas, as well as septicemia, which is a serious bloodstream infection. Being anaerobic, it can thrive in environments with little to no oxygen. Clostridium septicum can be found in various environments, including soil, water, and the gastrointestinal tracts of humans and animals. It can also be present in feces, wounds, and infections caused by other bacteria. This highlights the opportunistic nature of Clostridium septicum, as it can take advantage of pre-existing conditions to cause infections. Clostridium septicum is a large, rod-shaped bacterium with rounded ends. It is a gram-positive bacterium, which means it appears purple when stained with gram stain. It produces oval endospores that are located at the center of the cell. To culture Clostridium septicum, it is typically grown on blood agar or anaerobic media. These cultures are maintained under strict anaerobic conditions, meaning that they are kept in an environment with little to no oxygen. When grown on these media, the colonies of Clostridium septicum are usually circular in shape. One characteristic feature of these colonies is that they may produce gas bubbles. This is due to the fermentation process carried out by the bacteria, which can result in the production of gases as byproducts. Clostridium septicum is classified as a strict anaerobe, meaning it thrives in environments with little to no oxygen. It possesses various enzymes, including collagenase, hyaluronidase, and hemolysin. Collagenase helps the bacterium break down collagen, a protein found in connective tissues. Hyaluronidase aids in the degradation of hyaluronic acid, a component of connective tissues. Hemolysin is a toxin that can damage red blood cells. Additionally, Clostridium septicum is capable of fermenting glucose, a process that generates energy for the bacterium. This fermentation can also result in the production of gas as a byproduct. Clostridium septicum has the ability to infect both humans and animals. It is known to cause infections in various animal species, including dogs, cats, and cattle. In these animals, it can lead to a range of illnesses and conditions, depending on the site of infection and the overall health of the affected individual. It is important to be aware of the potential for Clostridium septicum infections in both human and veterinary medicine. Clostridium septicum is capable of producing several virulence factors that contribute to its pathogenicity. Collagenase, hyaluronidase, and hemolysin are among the virulence factors produced by this bacterium. These enzymes play a crucial role in allowing Clostridium septicum to invade and destroy host tissues. Collagenase helps break down collagen, a major component of connective tissues, facilitating tissue destruction. Hyaluronidase aids in the degradation of hyaluronic acid, an important component of the extracellular matrix. Hemolysin, on the other hand, is a toxin that can damage red blood cells. The combined action of these virulence factors contributes to the development of gas gangrene and other infections caused by Clostridium septicum. Clostridium septicum has the ability to develop antibiotic resistance through different mechanisms. One mechanism is the presence of efflux pumps, which are proteins that can actively pump out antibiotics from the bacterial cell, reducing their effectiveness. Additionally, Clostridium septicum can acquire antibiotic resistance through mutations in the target sites of antibiotics. These mutations can alter the structure of the target site, making it less susceptible to the action of the antibiotic. The development of antibiotic resistance in Clostridium septicum highlights the importance of using antibiotics judiciously and implementing appropriate infection control measures to prevent the spread of resistant strains. While Clostridium septicum infections are considered rare, they can indeed be life-threatening. Certain risk factors can increase the likelihood of infection. Trauma, such as wounds or injuries, can provide an entry point for the bacterium. Surgical procedures also pose a risk, particularly those involving the gastrointestinal tract. Underlying medical conditions, such as cancer, diabetes, and immunodeficiency, weaken the immune system and make individuals more susceptible to infections. In such cases, Clostridium septicum can take advantage of the weakened immune response and cause severe infections. It is important to be aware of these risk factors and take appropriate measures to prevent and manage Clostridium septicum infections in high-risk individuals. Clostridium septicum infections usually occur when the bacterium enters the body through a wound or another portal of entry. Once inside, it can produce toxins and enzymes that contribute to the development of infection. These virulence factors can cause tissue destruction, leading to the characteristic gas formation seen in conditions like gas gangrene. In severe cases, the infection can progress to sepsis, a life-threatening condition characterized by a systemic inflammatory response. The ability of Clostridium septicum to produce these toxins and enzymes is what makes it such a dangerous pathogen. Prompt diagnosis and appropriate treatment are essential to prevent the progression of the infection and its potentially severe consequences. The immune response to Clostridium septicum infections involves the activation of immune cells like macrophages and neutrophils. These cells play a crucial role in recognizing and eliminating the bacterium. However, Clostridium septicum has developed strategies to evade the immune system. It produces toxins and enzymes that can interfere with host immune responses. These virulence factors can inhibit the function of immune cells, impair phagocytosis, and disrupt the signaling pathways involved in the immune response. By evading the immune system, Clostridium septicum can establish and spread infection more effectively. Understanding the mechanisms by which the bacterium evades the immune system is important for developing effective treatment strategies and vaccines. Clostridium septicum infections can be diagnosed using various laboratory tests. Blood cultures are commonly used to detect the presence of the bacterium in the bloodstream. Wound cultures can be performed to isolate and identify Clostridium septicum from infected wounds. Imaging studies, such as X-rays or CT scans, may be used to assess the extent of tissue damage caused by the infection. In addition to traditional culture-based methods, molecular methods like polymerase chain reaction, PCR, can be used to detect and identify Clostridium septicum. PCR amplifies specific DNA sequences of the bacterium, allowing for its detection even in small amounts. These molecular methods can provide rapid and accurate results, aiding in the timely diagnosis of Clostridium septicum infections. Prevention of Clostridium septicum infections involves several measures. Good wound care, including proper cleaning, disinfection, and dressing of wounds, is essential to minimize the risk of infection. Practicing good hand hygiene, such as regular handwashing with soap and water, can help prevent the spread of the bacterium. Appropriate use of antibiotics, including following prescribed dosage and duration, is crucial to prevent the development of antibiotic resistance. It is important to note that currently, there are no available vaccines specifically targeting Clostridium septicum infections. However, vaccination against other Clostridium species, such as Clostridium tetani, which causes tetanus, can indirectly help prevent certain types of wound infections. It is always advisable to consult healthcare professionals for personalized advice on preventive measures and appropriate management of wounds to reduce the risk of Clostridium septicum infections. The treatment of Clostridium septicum infections usually involves a combination of antibiotics and surgical debridement. Antibiotics are used to target and eliminate the bacterium, while surgical debridement involves removing infected or dead tissue to prevent further spread of the infection. In some cases, hyperbaric oxygen therapy may be utilized as an adjunctive treatment. This therapy involves breathing pure oxygen in a pressurized chamber, which increases the amount of oxygen dissolved in the bloodstream. This can improve tissue oxygenation, as Clostridium septicum is an anaerobic bacterium that thrives in low-oxygen environments. Hyperbaric oxygen therapy can help inhibit the growth and spread of the bacterium, promote wound healing, and enhance the effectiveness of antibiotics. The specific treatment plan for Clostridium septicum infections may vary depending on the severity and location of the infection, as well as individual patient factors. It is important to consult with healthcare professionals for appropriate diagnosis and treatment recommendations. Clostridium septicum is indeed a rare but highly dangerous anaerobic bacterium that has the potential to cause severe infections. Its ability to produce toxins and enzymes contributes to tissue destruction and the development of sepsis, a life-threatening condition characterized by a systemic inflammatory response. Given the seriousness of Clostridium septicum infections, early and accurate diagnosis is crucial for timely intervention. Prompt initiation of appropriate treatment, which typically involves a combination of antibiotics and surgical intervention, can significantly improve the chances of a successful outcome. It is important for healthcare professionals to be vigilant in recognizing the signs and symptoms of Clostridium septicum infections and to promptly initiate appropriate diagnostic and therapeutic measures. This can help mitigate the potentially devastating consequences associated with this pathogen. Thank you so much for watching. If you enjoyed the video, we kindly ask you to give it a thumbs up and consider subscribing to our channel. Your support and engagement mean a lot to us, and it helps us create more content that you'll love. Don't forget to hit the notification bell to stay updated with our latest videos. Thank you again, and we appreciate your support.